Hi, my name is Logan James, and I'm a postdoc with Mike Ryan at UT Austin. And today I'll be telling you about evolutionary and allometric insights into auditory sensitivity and morphology of frogs. So a communication signal, of course, requires the coordination both within and between sender and receiver across time. So I study frogs, and so I think about here this male frog and how he has to have the correct morphology and neurology to produce his species' typical courtship call. And then I think about this female frog who has to have the correct morphology and neurology in order to receive and respond to that call. And that has to be coordinated again, both within and between these individuals across evolutionary time. One factor that can constrain this coordination is allometric scaling with body size that often has a large impact on morphology and neurology in invertebrates and other animals. So we wanted to leverage the frog study system, which has thousands of vocalizing species, in order to ask how this coordination occurs across time in relation to body size scaling. So we focus on the female frog here, or generally the receiver end, by looking at the frog hearing, so the auditory morphology and sensitivity here. So in this schematic, we see the incoming call, which we measure with the dominant frequency, which is important, in frog species, which first impinges on the tympanum, which is an external ear organ that we can measure the area of, which then sends its sound signals to two different papillae, the amphibian papilla, which has hair cells that respond to lower frequency sounds, and the basilar papilla, which has hair cells that respond to higher frequency sounds. Altogether, this morphology leads to some sort of auditory sensitivity, which we can measure based on a decibel threshold for a response from the frog. So what we did is took this schematic and we looked into the literature to find as much data as we could of these different features across species. Now, just to show you what that tympanum looks like, it's this pretty classic circular organ that you can see, especially on these large bullfrogs. Again, this external ear organ then sends the signals to the internal papillae, which have hair cells. Okay, so we looked through the literature to find these features, and we have two main questions. The first of which is whether these features do indeed scale with body size. We certainly suspect that they will, and they seem to, but from these measures of morphology and sensitivity, the correct phylogenetic controls haven't been used as far as we know. And then once we figure out that relationship with body size, can we observe any evidence for coevolution of these features beyond that expected by changes in body size? So here's a phylogeny of all the species for which we found at least one measure of auditory morphology or sound sensitivity. So you can see some of the larger groups of frogs like the hylids and the ranids here. And I'll be color coding the data in case you're interested in the relationships among these specific groups. The data mainly came for morphology from Fox 1995, and for auditory sensitivity, it came primarily from a table by Walt Wodzinski compiled in Taylor et al. 2019. So our first question, do these features of frog hearing scale with body size? Well, here we can see broadly the answer is yes. On the x-axis, we have the snout vent length, so a measure of the frog's body size. Then on the y-axis, we have various measures of the frog auditory system. So the dominant frequency here, we see that the larger frogs have lower dominant frequency calls. We see that larger frogs have larger tympanum areas. Larger frogs have more hair cells in both their amphibian papilla and their basilar papilla. And larger frogs are more sensitive to sound. So this is a little bit counterintuitive, but because this is a measure of decibel threshold, the lower the value here, the quieter the sound the frog responds to, so the more sensitive they are to sound. So larger frogs are responding to quieter sounds than smaller frogs. So again, we knew this relationship with dominant frequency, we've known it for a long time, but these relationships with sound sensitivity and auditory morphology hadn't been conducted as far as we know using these phylogenetically controlled comparisons. Okay, so yes, these features do scale with body size. So then our main question, once we confirmed that, was whether or not these features could co-evolve beyond the changes expected by changes with body size. And what you can first see here is that our sample size is unfortunately quite reduced. There's very few species for which we have both measures of auditory morphology and sound sensitivity, which is a little bit surprising and disappointing. But what you can see here when we look at the residuals with their correlation with snout vent length, On the x-axis here, we have positive points indicate species that have more hair cells in their amphibian papilla than you'd expect based on their body size. And on the contrast, on the left, we have fewer hair cells than expected. And then on the y-axis, we have frog species that are less sensitive to sound than expected by chance on the positive side, and species that are more sensitive than expected by their body size on the negative side. So this negative correlation here indicates that species that have more hair cells in their amphibian papilla than you'd expect based on their body size are also more sensitive to sound than you'd expect based on their body size, indicating that AP hair cell counts and sound sensitivity may be co-evolving independently from body size. Another interesting result we found was that the tympanum area here, if they had a tympanum area that was larger than expected based on their body size, the call also tended to be lower in dominant frequency. And this makes sense because we expect that larger tympanums should be able to hear lower frequency sounds than smaller tympanum areas. Okay, so overall, what did we see? We found that yes, indeed, these features do scale with body size, which is, again, not necessarily super surprising, but important to confirm with the correct analyses. And we also see some evidence that these features can co-evolve beyond these changes with body size. So again, there's some small sample sizes, but we do see that larger tympanums for their body size tended to co-evolve perhaps with lower frequency calls, and we found that more AP hair cells for their body size tended to have better hearing for their body size. And like I said, it'd be great to get more data, especially with this low overlap between measures of auditory sensitivity and auditory morphology, which of course is even more important now as we see the numbers of species of frogs declining rapidly around the world. So with that, I'd like to thank you for listening. I'd like to thank Mike Ryan, my supervisor, and Kim Hunter and Ryan Taylor, my collaborators from Salisbury University, as well as Rachel Page, who helped get funding for this work, and Walt Wazinski, whose ideas really were the impetus to start this project. Thank you so much for listening, and I'm happy to take any questions. My email and Twitter handle are on the left. If you'd like to contact me that way, that's fine as well. Thank you.